This is Forrest, and I'm presenting about Mycobacterium leprae. This is the bacteria that causes leprosy, or Hansen's disease. It's an obligate intracellular parasite. It cannot be cultured. It's characterized by very slow growth, prefers cool parts of the human body, and is a very close relative to Mycobacterium tuberculosis. It's part of the genus Mycobacterium, phylum Actinobacteria. It's gram-positive and has high GC content. This bacteria is between 1 and 8 micrometers long and about 0.3 micrometers in diameter. It's rod-shaped, can be slightly curved or straight, and has rounded ends. It does not form endospores. It's gram-positive. Importantly, its cell walls contain mycolic acid. This impacts many of the characteristics of this bacteria, including its very slow growth and its ability to live within phagocytes. It also requires a different kind of staining technique. It also has a capsule, and it very closely resembles Mycobacterium tuberculosis. In terms of metabolism, it is aerobic. It takes about 12 days to double, which is very slow. It grows best at about 30 degrees Celsius, preferring cooler regions of the human body, like the skin, peripheral nerves, eyes, upper respiratory tract, anything that's cool. It does generate its own ATP, uses glycolysis, citric acid cycle, but it depends upon its host for a large part of metabolism and for nutrition, and has extremely reduced the genes in its genome, preserving only what it needs really to survive, relying on the host for the rest. It does have a robust lipid metabolism, though, and can synthesize many types of lipids. I did not find much information about the interactions of this microbe with other microorganisms. However, it greatly interacts with its hosts, mainly humans and armadillos. It primarily targets Schwann cells, which wrap our nerves and help conduct nerve impulses, and macrophages, part of our cell-mediated immune system. Interestingly, it's the only known pathogen capable of infecting human peripheral nerves. How bad the presentation of leprosy in a person is depends in part on how many bacteria are present in the tissues. There's a spectrum of the disease ranging from paucibacillary to multibacillary. In PB, there's just a few bacilli in the host tissues, and the person's body had a strong immune response, and leprosy or the disease never really got a strong hold in the body. Whereas in multibacillary disease, over many years there can be up to billions of bacilli in the host tissue, and this happens when there wasn't a very strong immune response to the bacteria in the beginning. The symptoms of leprosy are going to present differently based on how advanced the disease is, but in skin, you're going to be experiencing numbness, discolored patches, rash, growths, bumps, ulcers on the feet. In terms of nerve damage, again, numbness, weakness, paralysis in more advanced cases, nerve enlargement, red areas, swollen areas, eye problems, blindness. In advanced cases, we're going to be seeing the reabsorption of extremities. The skin is not decaying or dying, it's being reabsorbed by the body. Loss of nasal tissue, blindness, disfigurement, paralysis. It can be very serious, but only after many years. It takes many, many years to develop to this point. Despite how feared this disease has been for thousands of years, and how disfiguring it can be, it actually takes years to develop this disease, and it takes a very long time to actually catch it. It's not very virulent. It can be treated, and it's not very difficult to treat. You just have to have access to treatment. You use a multi-drug therapy, multiple antibiotics, and it takes 1 to 2 years to complete. If you don't have the very advanced disease, it is treated with dapsone and rifampicin, and in an advanced case, it's treated with dapsone, rifampicin, and clofazimine. This was such an interesting organism to study, and there's so much more to say. It's also a very difficult organism to study because it cannot be cultured. Thank you for listening to my presentation.
Neisseria gonorrhea is a gram-negative bacteria and part of the Neisseria genus. It's one of two pathogenic species of the genus, and it causes a sexually transmitted infection, gonorrhea. The bacteria only impacts human, and as an obligate human colonizer, it cannot survive outside the host. In the United States, gonorrhea, along with chlamydia, are two very common STIs. There continues to be increasing rates in both. Management is challenging because many infections are asymptomatic and antimicrobial resistant strains of gonorrhea continue to develop and change. The shape of Neisseria gonorrhea bacteria is diplococci, and they are held within a polysaccharide capsule and lipogalactosaccharide cell wall made of lipid A. This layer enables the bacteria to attach and invade human cells. Other virulence factors that enable adherence to human cells include fibria, which are rod-like extensions. The bacteria also has pilus, which are a special type of fibria that are fewer and longer and enable the transfer of DNA to other cells in the process called conjugation. The pili specific to Neisseria gonorrhea, not pictured here, is a type 4 pili, which allows for the creation of microcolonies on the epithelial cell surface. They also have highly variable surface antigens, and this prevents the development of immunity and effective vaccine. The bacteria can infect the mucous membrane of the mouth, throat, eyes, and rectum. Sexual contact with the penis, vagina, mouth, or anus of an infected partner is the main way it's transmitted. It can be transmitted during childbirth and infect the infant. Neisseria gonorrhea grows after transmission, and the contact is established with the mucosal epithelia. First, the bacterial must adhere to the epithelia of the mucosa with the help of the type 4 pili and the lipopolysaccharide, LOS. Once that is complete, the pili help establish microcolonies. Diagnosis, treatment, and prevention of gonorrhea prove complicated, as it's a very common STI and many people do not seek screening because they are asymptomatic. Regular screening is therefore important, and samples and specimens can detect the presence of a gram-negative diplococci. Once detected, treatment is tricky too, because the widespread gonococcal strain resistance to antibiotics. Because the bacteria have highly variable surface antigens, as mentioned before, immunity doesn't often develop, and if it does, it doesn't often have broad protection. The impact of infection can be both symptomatic and asymptomatic. In men, symptoms can be acute inflammation and infection in the urethra causing painful urination. Men can experience an asymptomatic infection. Women are more likely to experience asymptomatic infection. However, if they do have symptoms, it could look like dysteria, increased vaginal discharge, or vaginal bleeding between periods. Although women may not experience symptoms, the infection can lead to additional conditions such as pelvic inflammatory disease. This happens when the infection travels by bacteria attaching to the sperm via the fimbriae or the LOS to the uterine tubes and cause inflammation, which can lead to ectopic pregnancy after scarring or infertility. Disseminated gonococcal infection, DGI, can happen if gonorrhea goes untreated and spreads to the bloodstream. Consequences of DGI include arthritis, dermatitis, tannovitis, and the condition can be life-threatening. Considering many people do not experience symptoms, this is a very dire consequence. Because so much of gonorrhea is asymptomatic, regular screening is essential for management. This visual shows the history of discovered and recommended antimicrobials and the evolution of the resistance in Neisseria gonorrhea. As you can see, from 1990 through to 2020, there have been numerous antimicrobials that have become no longer active. With the ever-changing landscape of antimicrobial resistance, prevention, screening, and treatment are essential. It is a major public health concern globally.
For my presentation, I'll be talking about Clostridioides difficile in healthcare. C. diff is a rod-shaped, gram-positive obligate anaerobe capable of producing toxins and endospores, and it's commonly found in healthcare facilities such as hospitals and nursing homes/assisted living facilities. Those with C. diff infection or CDI will have a altered gut microbiota, which can result in nausea, diarrhea, sepsis, and even death. There are others like C. diff that produce similar symptoms, such as Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, and Clostridium perfringens, which causes gas gangrene. So for those who are at risk, it will most likely be geriatric patients who are often found in hospitals and care homes and other living facilities. And antibiotic regimens are also a risk factor since antibiotics tend to alter the microbiota of the human body, which can leave a person with a weaker defense system against pathogens. In order to understand how C. diff works, it's important to look at its life cycle, composed of 4 different stages. And the first stage is called germination, and it starts by germinating and seeing if their environment has the appropriate nutrients necessary for growth and survival. And C. diff is pretty flexible with its nutrients, but some are necessary for metabolism, such as leucine, glycine, and proline, which are amino acids. The next stage is called the vegetative stage, and it's called the vegetative phase because once attached, bile acids and cogerminal signals to start growing, and it partakes in several types of metabolic pathways, such as Stickler metabolism, in which electrons are transferred from one amino acid to another to produce ATP, and metabolism has to be regulated, and in C. diff, it's regulated by the catabolite control regulator CCPA. And it regulates several metabolic pathways and even controls genes that release toxins, TCDA and TCDB, that C. diff uses to infect people. And CCPA indicates a link between metabolism and toxin production. The third stage is called toxin production, in which it occurs in the absence of nutrients, which causes C. diff to revert to its spore state, which sets it up for its final stage, which is called sporulation, in which it expels itself and waits for its next available victim. So there are a couple of microbes that interact with C. diff and its life cycle, and one of them is called Bacteroides thetiotomicron, and it produces sialidases, which promotes the growth of C. diff. There are a couple of microbes that actually inhibit C. diff, which are lactobacilli, such as L. gasseri and L. acidophilus, and since they produce an acidic environment, it inhibits the growth of C. diff. Clostridium scindens also produces antibiotic compounds, which inhibits the growth of C. diff. So, precautions and safety methods. The most important ones will be frequent hand washing and to only use antibiotics when necessary, since they can alter your gut microbiome. Another important one is to practice precautions in hospitals. It's pretty common practice to have an indicator outside of a patient's room stating that they have a C. diff infection, and so when somebody is a provider and needs to care for that patient, it gives them an idea of what to wear and what to do when caring for that patient so that they can protect themselves.
Organism is cyanobacteria. So 2.4 billion years ago, this planet had no oxygen, which meant that it had no life, what we would consider life on it. And one of the microorganisms that was kind of like floating around, there was a change to its genetic makeup or a mutation that gave it the ability to use photosynthesis because there was sun. And because it was able to use photosynthesis now, it was able to produce oxygen, which was something that this planet had never seen before. It was kind of like the jumpstart to life on the planet. And it was cyanobacteria that was that organism that was able to use photosynthesis. Bacteria is also known as a blue-green algae or bacteria, and it's usually found in water, so like rivers, lakes, oceans, seas, but it has been found in soil as well. Its most common shapes are spheres, rods, and spirals. It was originally known as an algae because its ability to use photosynthesis, but because it lacks qualities that other algae and plants have, so it does not have a membrane brown nucleus, and it doesn't have chloroplast, so they reclassified it into the prokaryote, and it is one of the only prokaryotes that has the ability to use photosynthesis, though it's not the only one, and it is also gram-negative. Cyanobacteria is made up of 4 different layers: the cell membrane, a peptoglycan layer, an outer layer, and a sheath. And its color comes from chlorophyll, which is responsible for the green color, and the phycocyanin, which is responsible for the blue. And it's the phycocyanin that's also responsible for its ability to use photosynthesis because that's what absorbs the sunlight. It needs a few things to survive: water, carbon dioxide, sunlight, and other nutrients. Most importantly, nitrogen and phosphorus of those other nutrients. It does tend to borrow electrons from whatever water it's around, and it kind of like disperses everything and releases oxygen into the atmosphere. But a fun little fact, if the cyanobacteria does not have sunlight or carbon dioxide, it can still survive if and only if there is enough glucose somewhere for it to kind of absorb or feast off or eat, then it can survive without having any sunlight around. Spring and the summer, whatever water it's around, there's usually higher levels of phosphorus or nitrogen, especially if it's like stagnant water, and it kind of will feast off of that in abundance and start reproducing, and it uses cellular fusion, so the parent cell splitting into 2 identical daughter cells, and it'll do that in rapid succession if there's enough nutrients around. And when that happens in quick succession, it's known as cyanobacteria bloom. So the cyanobacteria blooms are what you would be able to see out in nature without the assistance of a microscope. Usually it's a pea green color. It's very slimy. It's musty. It does grow big enough to where it will block sunlight from other organisms in its vicinity. It will, like, eat all the nutrients that other organisms may need. It does produce toxins as well, which is known as the cyanotoxin. And if a person comes into contact with the toxins produced, you can either touch it with your skin, you can breathe it, you could swallow it. whatever symptom that you may end up having, they'll usually appear within a 3 to 5 hour window from exposure and usually last a day or two. But these toxins can be deadly to other organisms, so like other plants, animals, and stuff like that. After seasons change from like spring and summer with the blooms and everything, it will start to die down during the autumn and the winter months because the environment's changing, there's not enough nutrients and everything, so it does produce endospores. So when everything's pretty much exhausted, it will release the endospores, and the endospore, depending where it's at, will like bury itself in the ground and just waiting for the environment to be more hospitable for it to start the cycle again. That was it. Thank you.
My favorite microorganisms are a group of bacteria known as Rickettsia. They are one of 4 of the genuses of the family Rickettsiae. They're typically rod-shaped gram-negative bacteria. This group of bacteria are known as obligate intracellular parasites, which means that they are organisms that absolutely require eukaryotic hosts to survive and replicate. Aspects of their structure that are notable include their lipopolysaccharide outer membrane and a surrounding slime layer. These organisms are also non-motile. Because they are obligate intracellular organisms, they depend upon eukaryotic host cells, like those of arthropods, like fleas, ticks, and lice, that act as hosts and also reservoirs for this type of bacteria. These arthropods act as vectors for transmission of this bacteria into human or animal hosts, where they then live in the cytosol of host cells. For their metabolism, they import or steal important biomolecules from their hosts to reproduce and metabolize. With regard to reproduction, they do so through binary fission, and in regards to metabolism, they perform aerobic respiration, although they do lack the glycolysis process. Rickettsia are also able to metabolize ATP using glutamate from their host. They also have adapted complex transport mechanisms to help fulfill their metabolic and reproductive needs, since they depend on these eukaryotic host cells. These 3 microorganisms reflect those notable in transmission of human disease with regard to Rickettsia bacteria. Starting with Rickettsia rickettsii, this microorganism is known to cause Rocky Mountain spotted fever in humans. Hard ticks act as the vector for this bacteria, where they enter endothelial cells from the human bloodstream. They cause disease by leaking blood into other surrounding tissues. Rickettsia prowazekii is known to cause epidemic typhus in humans. It is brought on by human body louse. These lice defecate the bacteria onto the host, which would then enter through scratching the bacteria into a skin wound. In some cases, this disease can reoccur in those who have had it previously, and it is known as the Brill-Zinsser disease. One unique trait about Rickettsia prowazekii is that it actually kills its arthropod vector, which deviates from the standard notion of symbiotic relations. Rickettsia typhi is known to cause endemic or murine typhus. It is transmitted through bites of rat or cat fleas onto human hosts or rodent reservoirs. This disease is rarely fatal even if left untreated. It also has other portals of entry that include the respiratory tract or conjunctiva. The 3rd form of typhus, known as scrub typhus, is caused by a microorganism of the Orientia genus, which was formerly a part of the Rickettsia genus. It is caused by red mites as a vector, which bite onto human or animal hosts. This microorganism acts parasitically towards its mite vector through manipulation and control of reproductive and cellular processes, which again deviates from symbiosis. Another important interaction between arthropods and Rickettsia bacteria includes the way in which infected arthropod vectors are maintained throughout generations. This occurs through transovarial passage from infected females to their ova, which then grow into more infected offspring. This is a primary way for various arthropod species to continue the infection of rickettsial diseases.
Hi, I'd like to introduce you to my favorite microbe, Salmonella. This tiny microscopic organism lives in the guts of animals like poultry, reptiles, cattle, and other farm animals. For a time, it can also live in soil, water, and on inanimate objects. Only 2 species exist in its family, Salmonella enterica, which has 6 subspecies, and Salmonella bangori, which is exclusively found in reptiles. While there are just 2 species, there are nearly 2,600 strains currently accounted for. While it may be tiny, this bacteria packs a powerful punch. If food or water contaminated with the bacteria are consumed, humans and other animals can become quite sick. But more about that later. Let's dig into who this tiny little microbe is. Salmonella is a bacillus, or rod-shaped bacteria, with 5 to 10 long rope-like tails called flagella that help it move around the environment. It also has shorter protrusions called fimbriae that help it adhere to or stick to other cells and tissues. Special types of fimbriae called pili can actually transfer information from inside the cell to other cells. This is called conjugation. All of these spindly hairs are attached to a capsule that protects it from drying out and evading immune cells that might try to destroy it. Underneath the capsule is a cell wall made up of 2 main parts, a thin layer of sugars and amino acids called peptidoglycan, which is below an outer layer made up of various fats, proteins, and sugars. This construction classifies the cell as gram-negative. When it's stained to be able to be seen under a microscope, it appears pink. Like other bacteria, Salmonella have genetic information called DNA with 1 chromosome that carries information for how to grow, reproduce, and function. One thing this bacteria has that not all others do are plasmids. These circular pieces of genetic information outside of the DNA often provide details for how to be better at infecting hosts, and through conjugation can be shared with other bacteria. Maybe not everyone has heard of Salmonella, but many have heard of their close relative Escherichia coli, or E. coli. Like Salmonella, it's a proteobacteria from the Enterobacteria family, or enteric bacteria. Also, like Salmonella, some strains of E. coli are infamous in causing food poisoning. These 2 bacteria actually come from a common ancestor from over 120 million years ago. E. coli actually stayed a relatively good actor. Most of its strains don't cause problems and are found as part of our normal bacterial playground in our bodies. Salmonella, on the other hand, evolved to be more pathogenic, or illness-causing, and really excellent at infecting. While the 2 microbes share 90% of the same genetics, much of their differences are for things that give Salmonella a leg up in the game of causing infection. Salmonella is really successful at life. It can live in as wide a ranging of places as the inside of animal guts, come out in its poop, get washed into water, and live there for up to 30 days. How does it do that? Well, adaptability is key. For starters, they're facultative anaerobes. They can live with or without oxygen. While they thrive on glucose, a sugar, they can get by on many other nutrients as well. And while they prefer a balmy 98.6 degrees in a fairly neutral environment in terms of acidity, they can get pretty cold, pretty hot, and a little acid won't do them much harm. So basically, if you were a microbe and could pick one friend to live on a deserted island, Salmonella would be a pretty good choice. That is, unless you're Proteobacterium carnivororum, because there you are, sharing space on some greens on your deserted island with your pal Salmonella, eating away, and through your eating, you're actually creating nutrients for Salmonella to eat up. And how does Salmonella return the favor? They eat the nutrients you provide and make the plant environment so acidic you can't even stand to be there anymore. What a parasite. And not just a parasite, a pathogen that can really wreak havoc. That adaptability we talked about makes it so Salmonella can show up in a lot of unwelcome places, including our food. If we consume contaminated food or water, depending on the strain present, we can get really sick. Salmonella has 2 general types of disease-causing strains, one that causes enteric fever and one that causes salmonellosis. Enteric fever, also known as typhoid fever, is more common in lower-income countries where sanitation infrastructure may be underdeveloped. It's actually life-threatening, causing a fever, coughing, loss of appetite, and stomach ache. Diarrhea is often present too. It can be treated with antibiotics, though resistance is on the rise. For pathogenic strains that don't cause enteric fever, animals essentially get classic food poisoning symptoms. And for the humans in the United States, salmonella is one of the top 5 germs that causes food poisoning. Because of how our food is produced, salmonella can get into milk, onto eggs, on chicken, ground up into meat, and even on fresh produce. Symptoms of salmonellosis include stomach cramps, fever, and diarrhea. Unlike enteric fever, it's self-limiting, so most people don't take antibiotics. And what does self-limiting mean? Well, the diarrhea it causes does a really great job of flushing salmonella out of your intestine. Don't fear, though. Simple steps can be taken to avoid impacts. Thoroughly cook your food, wash your produce, and always wash your hands after you use the bathroom and before you eat. With a little care, we can avoid salmonella's negative impacts and marvel at our favorite microbes' ability to succeed at life.